The splice and clip plus tube technique is modified from the tube or slant cut grafting, which is the industry standard for propagating tomatoes. It involves making identical angled cuts on both the rootstock and scion, then joining the exposed stems together using a small flexible silicone or rubber grafting tube. For the modified splice clip plus tube tomato grafting, we need clips, razor blade, and self-made parafilm tubes, not commercial silicone tubes. We cut the parafilm into 1-inch strips and then cut the strips into 3 equal-length pieces. Each piece can be rolled into a flexible tube, which can be adjusted based on the thickness diameter of the tomato stems. Here is how we make tomato homografting, which is to join the shoot, scion, and rootstock of tomato plants that belong to the same accession or variety. We first break the razor blade into half and use one of them for grafting. We cut the tomato plants below the cotyledons at a 45 to 60 degree angle. The homografting serves as our control for heterografting experiments. Because the same accession will be used as scion and rootstock, we can cut the plants and put the shoot into sterile water to avoid wilting. We can easily handle 18 plants at a time. We use a small piece of stick to roll the parafilm into a flexible tube and put the tube halfway over the severed cut of the rootstock. We then slide the cut end of the scion into the top of the tube until the cut surfaces are pressed tightly together. We use a clip to hold the tube and the 2 connecting pieces. We can then continue to finish the rest 17 plants. Heterografting is to join a desirable tomato plant scion to a different variety's rootstock. Most commonly, this involves grafting tomatoes onto robust, disease-resistant tomato rootstocks. This time, we will perform reciprocal grafting. We first cut 10 plants of one accession and stick the shoot in sterile water. Then we cut the scion from another accession and finish grafting using similar process as homografting. After finishing grafting one tray, immediately cover the newly grafted plants with a dome cover. We spray water mist onto grafted plants and the inside wall of the dome. Carefully place the dome onto the tray to keep plants in high humidity, 80 to 95%. To ensure low light, we further cover the dome with a piece of black plastic sheet and then put the tray into a growth chamber. Grafted plants were kept in the dark or shaded environment for the first 3 to 4 days to minimize transpiration. Check the plants and spray mist onto plants if necessary. After that, gradually introduce the plants back to weak light and slowly decrease the humidity, eventually back to normal condition over the next week. The plants will heal in about 10 days.